Hi, welcome to this coding session in which we will be using a publicly available dataset from the BCI competition. It's a BCI competition IV and it's the first dataset, and you can of course see the publication that was related to this dataset. Um, it's a very nice dataset in which there is data of motor imagery, so people are basically imagining movements of right hand, left hand, and also uh, feet. So I'm just using some very small samples, like some participants in this dataset. By the way, some of them were artificially generated, but this is really irrelevant in this kind of coding sessions. So anyway, these are actual data. These are actual data sets that we will be using in this session. And as we said, we will be using EEG signals or electroencephalograms as our multivariate data, and we will be using some very nice functions from FieldTrip, and also we'll be using functions from EEG lab in the future, and we will be using functions from MVPA, which is another Matlab package. So we will be discussing them in the future, but right now we will be using some functions from FieldTrip and see how we can use it and add some other functionality in it inside our package Lively Vectors. I will be leaving links to like the dataset if you would like to download it and test the code yourself and play with the code. I really like to explore with this, and I really encourage you to explore with changing the code and seeing the result yourself. So okay, so we start this by just clearing our variables, so we can clear all the variables that we have. So let's say that we have a variable here. If we go to this workspace, we can see that we have x defined in our workspace. If we would like to clear our workspace, we can execute this. So I'm pressing F2 as we discussed in the other video, and we are clearing our workspace. And you can type in CLC to clear the command window. And okay, so we go to defining our participants. So basically, let's execute this code, execute this line, and what this does is defining uh, these names, these characters here. So each code is representing data from a different participant, and that's this character will be appended to our like file path, and then we'll be able to access different datasets. So here is another step. You can press Control D to open this variable, any variable that you like. So I can press Control D here and it will open this, and MATLAB will show you that this is a cell variable. So we are defining 5 cells. Each one of them is containing a value, a character that we will be using. Okay, so let's go to the other one, and this is the location where we put our datasets. So of course you are gonna have to change this to the location you have downloaded the datasets. And okay, so here I'm starting to place, as I said, something very important, which is the breakpoint, and then I will be able to press F1 to just start executing, and we will be stopping at this line of the breakpoint. Okay, so here we're just looping over the participants, so we'll be creating this loop to loop over them, and here I'm loading the data. As you can see, we here are loading our files. They are .mat files, And the nice thing in Matlab that you can concatenate a string or a double or whatever by just using these brackets right here. So basically each one of these is a string or um, a part of our path that we need to use. So this is the B that is going to be appended to this string right here. So, and then we append mat. So when we put it like that, it will give us the full path to this dataset. Okay, now that we are ready to continue with the data analysis, we have loaded our data and we can check our workspace to see that there are some new variables like NFO and MRK and CNT. And of course, you can go ahead and read the description of the dataset, or actually, I'm gonna check the description of the dataset in the video as well, such that we know exactly what was happening with the task. But anyway, we can actually stop at this line. So we're at 14. We can go ahead and check our dataset. So basically, feel free to stop the video or check the link that I put for the dataset. And basically, we can just look at this and see how the task looks like. And actually, we can like think about, let's say, try to visualize it like that. And I think it's a nice way to try to always visualize the study design, like the experimental design beforehand, instead of just reading the text all the time, like every time you need to update something. So this is saying that there was 4-second period and these periods were interleaved with 2 seconds of blank screen. Alright, so I think it was like looking like this. So they had like a 4-second period, and then they had 2 seconds like that of blank screen. So that's a blank screen, and then there were 2 seconds of fixation cross. So we can put it here. So that's 2 seconds. Okay, and I think this is it. So it's quite nice to have this task, and because it's clear what they are doing, and it's also available for free, so you can download it and exit it immediately. So there is a fixation cross here, and I think it was repeating. So I would be representing the onset of the cues, or like what is the external stimulus that was appearing to participants by this arrow right here. So this would be our time zero, where the stimulus were presented, and we can imagine that this will be repeating, like we will have another stimulus here, time zero, and this whole thing will be repeated here, like we'll have 4 seconds here, and we'll have 2 seconds here and 2 seconds there, unless of course there is a pause between different trials. Okay, so we can think of what is really interesting in this part is that let's imagine that we are trying to analyze our EEG data, and we are trying to see the difference between the motor imagination of left hand, what what will be the response of the brain for moving left hand or imagining to move left hand compared to moving right hand. So that's exactly what we are trying to see with this data set. So basically, participants are doing the motor imagination for 4 seconds. So we can imagine that if we have the onset like a marker for the beginning of the trial, if we know which sample is time zero, we can just cut 4 seconds after this onset and analyze this part, and cut another 4 seconds after a different cue and analyze this part. So imagine that if this is for right hand and this is imagination for left hand, we are trying to see the differences between the two. So we basically trying to see, would the signals of right hand somehow look like this and left hand will look different like this, such that we can later on look at the differences between the signals or do some fancy analysis and do some machine learning, uh, like training and testing, to pick up the differences between right hand and left hand imagination, or what we can do with this. So that's the whole story, very very quickly, very briefly. But the important thing is that we are analyzing this 4 second period, and we can also analyze these or use them as baseline, as we can see. But the important thing is now we have the dataset. We can look again at the code. So in the code we have stopped at this line 14, and we want to continue. So basically we can see that if we press Ctrl D and open this NFO, we see that it's a struct, and I have to say this data type will come in a lot of videos because it's very nice to have, and I think it's a bit similar to dictionary data type in Python. But anyway, it's very nice to have this struct, because with the struct you can define different data types inside the same variable. So let's put another thing inside of here. So let's say nfo.x, which is not here, equals 1 to 10, and you can see that x is now added to this, so it doesn't have to be the same type. And I think this is very nice to have, because if you have a struct representing your EEG signals, you will need to define a lot of things about the EEG signals. All of them are not of the same data type. Like if you are putting the name of the sensors, you wouldn't expect this to be of the same data type as the microvolts or the amplitude values of the actual signals. So it's just nice to have this data type and put all the information of your signals inside. So okay, so NFO, we get the sampling rate, we get the classes, so left motor imagery and right motor imagery, and in this one we have the labels of the sensors. So in this task they got 59 sensors, and we get the names of the electrodes. So of course each one of them is placed in a different place. Like for instance, this one is frontal electrode, and it's placed on the left side, because this value is odd. When it's even, it's the right side, and yeah, and so on. So feel free to check like a head cap, EEG head cap, and see the places of every sensor of these. And I think these are the X and Y position of the electrodes. So I think, yeah, that's nice. So here in line 14 we are extracting the first important information of this data, which is the labels of the sensors. So I'm just changing the orientation of this. I just need this to be a column vector. So now I'm putting this inside another struct that I'm defining, which is called dat. So now that dot label that we define will include the labels, and it will be column vector, so it will look like that. And here I'm defining the data itself, so I'm putting this inside dat.trial, and this will be like a continuous recording. So it's like the whole data set inside of here. So it was inside CNT, and we multiply by this to have it in microvolts, and I'm putting this inside dat.trial inside of this variable right here. So we'll have a struct, and we will have the trial, and inside of that we will have this 1 by 1 cell. So if we access that to trial, it's 1 by 1 cell, so just one cell, and inside of this cell we have this huge matrix of the whole recording. And by the way, this is similar to what you will be recording in Brain Vision or any like recorder and Brain Vision recorder or any other recorder, because 59 is our channels, so that's channels, and then that's samples. So we can say that this is uh, a channel by time, or a channel by samples. And then here we're defining the sample info. So basically I'm just taking the size of our second dimension here. So it's like the whole samples, and starting from the beginning, just to have an idea of our samples, like the beginning and end sample. And here I'm getting the sampling rate, putting it inside of here. And this one I had to have like a time vector. So I wanted to do a transformation from just having the samples and have an actual time vector. So our time vector will be starting from 0. So 0 will be the first time point, so it will be basically this whole column, because it's this the first uh, time point. And with this analysis we will see that, or any other analysis we will see that, if we go to the second time point, we are increasing by basically 1 over the sampling rate, so that if we take steps by 1 over the sampling rate up until the point we reach the end sample. The end sample, of course, is divided by the sampling rate again. We will be having a vector of time representing the actual time of each sample. So let's go ahead and execute this and have a look at it. So that the time will be a cell, and it will be of the same length. By the way, you can double click here and just copy this, which is a handy tip as well. So I can just copy this like that and say, okay, let's check the length of this, and the length of that is exactly equal to the size of our data. Okay, so I guess this is nice to have. We can also close all, so we can do this. I can close all of these variables and just have this variable open, and we can go to the next section. Okay, so in this section we will be looking at filtering our continuous data. And I have to say I have a lively vector script for segmenting our data and handling data from Brain Vision Recorder, of course with the help of FieldTrip. So it's a different script that I will be discussing later on, but it will basically enable us to uh, see how we can load big data sets. I think this is important, because if you have a huge recordings and you have a data set, let's say from sleep, and that's what I'm actually analyzing at the moment, you will have a harder time loading your data set at once. So you need to load your data set in a different way, and I will show you how to do this, and how also to use FieldTrip uh, filtering methods to analyze your data that's inside our package. So basically what I'm doing here, because our data set in this example is simple, I'm just using some already existing filtering directly, and the dataset here is already filtered, so we don't need actually to filter the data. So I'm just including this if you would like to see how it looks like in FieldTrip and how we can use it. So we can define our struct, we call it CFG, and we can see that um, we can define it empty, and we can put some specs to it. So that's our like configuration struct. So we can do bandpass filtering and say yes to that. We're doing bandpass filtering. We can put like the beginning and end of our frequency band of interest, like from 0.1 hertz until 50 hertz, and we can do stop band filtering to get rid of like the line noise. Line noise are normally around 50 hertz, so by doing so we are getting rid of this kind of noise, and we can demean our data. What this means is that if we imagine that our data is looking like that, so if we have our signals looking like this, what dmean is doing, and of course this is continuous, so this is over time, and I'm just looking at one sensor, let's say C3, any sensor, and this will be basically taking the average of all of these. So let's say if this is our scale, and this is, let's say, -3, like this point, and this point is like 3, and 0 is here. What this will do, it will take the average of all of these points right here. So we can say that this average, the mean of all of these time points will be -3. So they are looking more or less around -3. And it will be subtracting -3 from all of the samples. So from each sample we subtract -3. So what this will do, it will bring the mean to 0. So this will bring our mean to 0, which is nice actually to see. So we can say that our signal will be moved to have a mean of 0, and we will see why this might be important and nice to have. And yeah, so to continue with this, we can just go ahead and give this pre-processing function in FieldTrip. We can give it CFG, and we can give it our struct, and basically our struct got to follow a specific format. That's why I was just copying everything from the dataset inside of this struct to match the format of FieldTrip. So right now we are just pre-processing our data. I'm copying this from that inside that. So now our data is filtered, and it's the same data. But of course there are differences in the values because they are now filtered. Okay, now to the next part.